Hey everyone, my name is Sid Patel. I'm a director of digital medicine at DIME, or also known as Digital Medicine Society. And today I'm super excited to be a part of this conversation with you and share a little bit about developing clinically meaningful digital clinical measures. And something that DIME or Digital Medicine Society is a professional society, one of the first, where we bring people, professionals, people like you from all disciplines and all backgrounds. And our aim is to promote safe, effective, ethical, and equitable use of digital medicine to optimize health. So before we get started, imagine you're driving your car. You know, when you're driving your car, you remember and you can see so many vitals. You can see the speed of your car, how fast or slow you're going. You can see the fuel, the brake oil, everything. But when it comes to body, our body is this unique box of information that for a very long time we were not able to tap into. But now, with the use of technology, we are able to understand and learn more. So, what are digital clinical measures? Essentially, when we try to measure any health outcome, behavioral data, physiological characteristics of an individual's health using some kind of sensor technology or connected tool, and that's measured digitally, are essentially digital clinical measures. And there's so many. For example, the voice box is measuring the cough and respiratory conditions, wearable sensors that we have seen, the commercial ones that you might know as Apple or Fitbit, that measures activity, heart rate. All these opens up a new era of how we create new technologies and how we create new therapies for our patients. And where do we see the benefit of these digital clinical measures? Essentially, they are in three areas: clinical research, where we'll focus our main today, clinical care, and public health. And if we see on clinical research, we there are so many advantages. One, in accelerating the timeline and the cost, to the applicability, to even generating more accessible research. I actually want to walk us through one of the case studies. Meet Priyanka Agarwal. She is the director of digital health at MyCardia, and she has been leading this development of new therapies for patients with obstructive hypertrophic cardiomyopathy, also known as HCM. It's one of the rare diseases where she's not able, because of the clinical variation in the trials for the patient, she's not able to measure and have patients come into the clinic just thinking differently on can be utilized in a decentralized format. But there are a few questions that she wants to know. Something like, what's a normal activity level for healthy men and women who undergo HCM or hypertrophic cardiomyopathy? You know, what's their age? What do clinically, what is something that's clinically important for them? So at Dime, we created this small framework to understand and develop measures that actually matters for the patient. And it starts with identifying what's the meaningful aspect of health, what part of the disease that we want to know more about, what which is getting worse, how can we prevent it, how can we better it, and there are ways that we can critically have inputs from the patient. Once we identify the clinical meaningful aspect of health, we understand what's the interest out of that, which is a narrowed version of what are the symptoms that patients are going through a particular disease. And then we look at the outcomes that can be actually measured in a realistic format. And what are the specifications that are around? That leads us to a digital endpoint, essentially a precisely defined, statistically analyzed variable that a digital companion, a sensor tool, can actually measure for any disease. Going back to our case where Priyanka was actually trying to develop this new drug, she found she understands that for HCM, one of the key concerns is the ability to move or the activity levels because of the reduced blood flow, and that prevents patients from performing specific daily activities. Now she wants to know how can she measure an activity as a meaningful aspect of health, and where should she start from? So if we go back, activity is such a broad term. You know, there's a duration of activity, there's a frequency of activity, there's intensity of activity. Within duration, we can capture and measure minutes per day walking, we can measure cumulative daily minutes. So looking at all these parameters, how do we understand what's a good measurement process? So she created a novel measure by identifying she wants to have walking ability for HCM patients, where how she'll do it is by digitally collecting using an accelerometer or gyroscope using a wearable sensor. And what she will measure is accelerometer units, the units that a patient utilizes for walking. Boom. From sometime before, when she was identifying how to create digital clinical endpoint for a new rare disease, to here she's designing a novel measure within minutes. This takes a lot of time, and it takes an entire framework to make it successful. Um, what we talked about is just a small measurement component. They are components where we have to identify what's the right technology. How are you going to evaluate? How will we validate? What are security and usability and utility concerns that we need to do? And to make sure that the job is well done, we need to implement and plan our operations. And we lay out this entire framework in our initiative at Playbook, which I encourage you to check out at dimesociety.org, which is essentially bringing from experts and stakeholders from FDA to digital health companies to even our partner co-panelist Jen was a part of it, which we are very excited to have. So this is a resource, open source, you can tap into if you're looking for it. But then you would ask me, which digital endpoint is right for your research and how can science support you? Well, we have an answer. Looking at digital endpoints, there's so many of them, and it's hard to bring home to all these endpoints at one place. So what we did is we created a library along with experts where we identified and we have a resource for you. If you are looking for a specific activity and a specific measure, you can go in and identify what it would be utilized. So this is, again, an open source resource, and we encourage you to check it out. And just to give you a snapshot on till today, from October of 2019 to March of 2021, we are able, with the experts and sponsors who have supported through and shared what are some of the endpoints, we are able to identify 207 unique digital endpoints. And it ranges from utilization for investigational product of drugs, for devices, for biologics in various different clinical trial settings. Essentially, why does it matter? Why are we having this conversation today at this conference? I want you to meet Pat Palms. Before I knew about Pat has Duchenne's muscle dystrophy, where essentially it's affected for young men and young boys who are on wheelchair before they even hit the middle school, and their lifespan is very short. The gold standards of measures today exist, like 6-minute walk test, essentially breaks my heart because people like Pat, there are so many patients who essentially cannot, 60% of the population is essentially excluded. And it makes me think that, do we not involve them for our research? Do we not include them? People like Pat are the ones who need these resources and are the ones who wants to have the endpoints being developed for. And digital measures helps us to do that. And lastly, I'll leave with that we have to be as brave as our patients and our people need it. So thank you so much for listening today.
Hi there, I'm Jen Horangeout, the founder and CEO of Savvy Cooperative. Savvy's a little bit different. We are a patient-owned co-op that helps to connect companies and innovators and researchers directly with diverse patients and caregivers. And I care so much about speaking with you today and about this topic, about how we can collaborate to make better patient-centered endpoints, because I am a patient first and foremost. I was diagnosed with multiple autoimmune conditions when I was an infant, notably juvenile idiopathic arthritis and uveitis, so some rare, some common. I have a laundry list of other conditions as well, but those were my first two diagnoses. I also had a brain tumor removed about 8 years ago, so very entrenched in the patient experience. And I then went on to become a human factors engineer and human-centered designer as well as a PhD studying patient-centered outcomes prior to starting Savvy. So I've seen this problem in a lot of different ways, and I'm really excited to be able to talk to you today about some of the things that I'm seeing both on sort of the professional side, but also what I see as a patient, knowing what it's like to be going through a lot of these things, either with your own treatment or being part of trials, and how can we make it so that we're going to measure things that matter to patients? That's really what I want to do first and foremost. So to kick it off here, to give a little bit of this sort of context, wanting to understand, why are we talking about this? Well, what I saw, when I was working with my professional colleagues in industry and academia and also in regulatory bodies, that there was often this disconnect that happened between the professionals and the patients and caregivers. Everybody well-meaning, wanting to do the right thing, to be able to innovate the best way possible for patients. It just didn't have all that information. And it meant that we were creating things or bringing drugs to market after 15 years that didn't always solve the problems that were most important to patients and caregivers. And that's really what we want to be able to be here today and say, is there a better way that we can co-design the future together? And so that's what we do day in and day out at Savvy Cooperative, is to be able to connect researchers and innovators directly with patients and caregivers. Many of you are probably already doing this work, which is why you're here, or you're curious. You're at least, patient-centered curious. And I'm really excited to be able to just share a little bit about what I'm seeing and why this is so important. So, like I say, I'm a patient first and foremost. And as somebody in the rheumatology space, I can tell you about, some anecdotes that I saw either on a personal standpoint, but also what I saw in my peers and other people doing this kind of research. And of course, talking about what happens outside of that specific therapeutic area. And that's what I think is certainly in the rare space. There's a lot of commonalities that people are trying to repurpose drugs here, there, otherwise to be able to serve the rare population. But it's not a one-size-fits-all model. We really need to be able to do the work with each individual patient group to understand what their experiences are. And Smit gave a great example at the end there that, certain different ways that we measure things will not work for everybody, let alone for the different therapeutic areas. And so that's why we need to start by even just doing simple conversations with patients. What are their unmet needs? What is it like for them to live their lives and try to fulfill thriving lives? Obviously, we're not all just trying to, go to clinic visits. We want to be able to go out and work and play and be with our peers and friends. So trying to really just first and foremost get to the bottom of, what's your life like? What is it like? Let's have an actual conversation before you try to integrate, getting feedback on various different things like an endpoint. But before you start developing out that endpoint, my challenge to you all is, how well do you know the community that you're innovating for? The next thing is to really take a pause and talk about diversity and inclusion. Obviously, we need to be thinking through the diverse individuals that are living with a condition. And are we talking to them throughout that process? So I just said you need to do what the sign behind me says, ask patients. But we need to make sure that we're talking to a whole lot of patients that represent different experiences, different backgrounds, different priorities, because we don't want to just innovate for a subset of the population. We really want to be inclusive and innovate for all. So my challenge back to you all is make sure you're doing the work, you're setting aside the time and resources to really get to know the community as a whole. So now let's talk about what we're here to talk about, sort of the outcomes and the endpoints. So in academia, what I was working on was patient-centered outcomes, and how do we include what matters to patients in clinical trial outcome measure sets and in treatment guidelines. That's what we always talk about, shared decision-making in healthcare. But if we don't measure what matters to patients, how can we have that informed conversation with a provider or our care team? We need to measure what matters to them. And so I did a lot of work in rheumatology. And one of the things that we found early on was that fatigue was not often measured. But if you talk to patients, honestly, with any autoimmune condition, they will tell you that fatigue is one of the most important things to get under control. You can have your pain controlled, but it can be so exhausting just to live with an autoimmune condition. And so over the years, as we did work to prove, actually patients really care about this, it became integrated into the core sets of outcomes that are measured during these different types of inflammatory arthritis clinical trials. And so that was so so important. And I think if we start to now think, okay, what does that look like as we think about these digital endpoints? And I'd love to share a little story with you. It's a great example. In a rheumatology trial, in an RA trial, rheumatoid arthritis, they were actually studying, what it was like for a therapeutic to be able to decrease the pain that a participant had. And that was the hypothesis, right? As the treatment worked, that the pain level of the patient would go down. And they also thought that the pain would go down, activity level would go up. So they actually had a wearable, an activity tracker. And what happened was, for most people, that's what happened. But there was one person that they saw as pain went down, activity levels also went down. And so they called up that participant and said, hey, what's going on? Is there something wrong with the tracker? Are you not wearing it often? And they said, no, I feel great. My pain's going down, but I feel so good that I'm now able to go back to writing because I'm an author and that's what's most important to me. So it just goes to show that there isn't always a one size fits all. And even as we move to more digital endpoints, there may be things that we're not thinking about. And the only way we understand that is we actually work with the patients to think about, what are maybe some of those fringe cases to make sure we're not excluding them from being able to capture how powerful some of these therapies can be. And then that's something to think about too with remote trials. As we're using these different devices or ways of measuring things, what does that look like to make sure we're supporting patients in doing this? I found, especially in the pandemic, we threw all this stuff at patients to be able to support them remotely, which was fabulous. But at the same time, patients are juggling a lot of things. Does it integrate with what they're currently using with patient portals or how they're communicating with their other care team? We hear that all the time. How can my trial data also be shared with my current care team? So really thinking about these aspects, as well as do these digital endpoints, do they require Bluetooth and broadband? And how does that weigh into what a patient is trying to manage on their own in the sense of how are they paying for it? And are we being really thoughtful to make sure that our trials can be inclusive when we use these types of devices as well? But I think that we're really on the right track. And the best way that we can get there is to ask patients, and we can work together to co-create the future of healthcare, because I think we're going to get there faster and accelerate the entire innovation process when those innovators and researchers come together with patients and caregivers who are so passionate about changing healthcare. So thank you so much for letting me share a little bit about this, and I look forward to the discussion.
Hi, I'm Elizabeth Berry-Cravis from Rush University Medical Center, and I've been working in Fragile X syndrome and other rare neurodevelopmental disabilities for a long time. And I'm just going to share some of the challenges that we've had with endpoints for these disorders and some of the work that we've done to try to find endpoints that will measure things that are important to patients with Fragile X syndrome and other conditions. One of the problems in Fragile X syndrome is that we, for a long time, we used measures that the FDA kind of told us to use, like the Aberrant Behavior Checklist, which is abbreviated ABC. And that measures a list of behaviors that have been identified as problem behaviors in people with intellectual disabilities and developmental disorders. But that didn't always match really, really well to what the families were telling us were the real problems. So we recently did a survey to really find out, to ask the patients to really find out what the families felt was the problem in individuals with Fragile X syndrome, which is a neurodevelopmental disability associated with autistic behaviors and usually moderate intellectual disability, as well as a variety of other difficult behaviors like anxiety and ADHD symptoms. So when the survey came back, it was very clear that the drug treatment priority in Fragile X was for us to target anxiety, number one, and number two, cognition and learning, as well as a list of other things that were somewhat age dependent also. So we actually had we had a couple meetings with FDA several years ago and took this there. And the FDA felt you can't make anxiety a primary outcome in Fragile X because this is an internalizing symptom and the patients aren't able to self-rate. So we would have to use something like the Aberrant Behavior Checklist, which is a standardized measure for administration to families. Well, this is a problem because the families are telling us this is our biggest problem, and the FDA is telling us you can't do a trial targeted at that symptom. So the response of the Fragile X field, which has been going on over the past year or two, is to do a survey of the families. This is our kind of solution to this problem. We've done a big survey of the families asking, when you tell us the patient has anxiety, what do you see? And so we're now creating a multi-domain clinician global impression sort of scale that is looking at different domains of what the families say is anxiety. And so it's a measure of observable behaviors now, rather than something that has to be rated by the patient. And so this will then track to what the families told us was their quality of life limiting problem of anxiety. So this is one way we've approached this, and this is a work in progress, but it's just a way of attacking a problem that was kind of viewed as unmeasurable, but is really important to patients. Okay, so the second thing families told us is that learning and cognition is a huge problem for their family members with Fragile X syndrome. And when we look at cognition and intellectual disabilities, we can't even measure cognition in intellectual disabilities with standardized tests, because the tests aren't validated for that population. They're validated on normative populations. And if you get enough below the mean, then you simply sort of fall in this one big pot of the floor of the IQ test. So this is what this is what the Stanford-Binet looks like in Fragile X if you're trying to measure cognition. And this big group of patients just all falls at 40. And then there's sort of a little bit of skewing upwards. So obviously, even if we wanted to measure what was going on with cognition in a patient group with Fragile X, we'd have so much problem with floor effects that we wouldn't really be able to measure anything. So the solution was that when in great work done by David Hessel, we used a huge ID population coming from some of the big autism studies, as well as a lot of groups contributing Fragile X data, to modify the standardization of the Stanford-Binet for intellectual disability, including Fragile X, and using a Z deviation score, which looks at how, when you answer the items, how truly far off are you from the mean, or it's also called Windsorized scores, which give a true IQ. And when we use this measure, we can see there's huge variability in Fragile X, and we get this nice bell curve. And now we expect this score is going to more accurately predict meaningful outcomes in adults with Fragile X, ultimately, such as employment and where the patient is living and how they're functioning. But that's the Stanford-Binet, and it can't really be used as an outcome because it can't be administered frequently in clinical trials. So how do we measure cognitive change in a clinical trial of a person with intellectual disability? Well, the NIH Toolbox Cognitive Battery is a iPad-based measure that's been in development for many years in normal populations, because funded by NIH to produce a battery that could be used for cognitive testing across clinical trials in all sort of disease areas. Well, this had been nicely validated in normative populations, but had not been validated in intellectual disability populations. And this is just the different tests that are in this, and it covers a wide range of cognitive sort of constructs. And so we got an NIH grant to validate this test and to modify it so it could be done by people with intellectual disability, and validated it. And so particularly for the Fragile X group, we found that over 80% of patients could do the test and get a reliable score on all of the tests except for these three up here, which are more executive tests and are difficult for people with Fragile X. And so we also showed that there was good, very good retest reliability with these tests. If for the ones where the patients could do them, it was less good if the patients weren't so good at doing them, these three tests that are difficult. And we were also able to show that the test had good convergent, discriminant, and ecological validity, had a nice normal distribution, correlated extremely well with that IQ score, the Z deviation IQ scores, and in fact, even identified different profiles in groups with intellectual disabilities, such as differentiating Down syndrome from Fragile X syndrome based on performance on some of the executive tests. And then we ran a clinical trial of a new medication targeted to a mechanism in Fragile X, and showed that in this placebo-controlled trial, we could see improvement over placebo in the cognitive tests that are the four main tests that patients with Fragile X do well. And so this measure addresses the clinician's need for performance-based cognitive measure in intellectual disability for trials, and we're continuing to perfect it and develop better tests for intellectual disability in the areas that were more difficult. And it's currently, this is actually the best validated cognitive test in intellectual disability, because most cognitive tests haven't been validated at all for that patient group. But the FDA, we've presented this to FDA, and they want more validation, and they have actually suggested that we go back and use a quote standardized test, which is in fact a test that wasn't standardized in intellectual disability. So we have work to do on this, and we're trying to figure out what we need to do to be able to use this test, because otherwise this will delay trials of new mechanism targeted drugs in intellectual disability. So another problem we have in Fragile X and many rare diseases is that the standard measures, as we just heard, don't address the primary patient and family concerns. And if I can't even tell you how many times I've heard a Fragile X family say to me, I filled out all these forms, but they never asked me anything about his biggest problems. And of course, this is the issue, that the forms didn't necessarily talk to the families with specific diseases, where we want to have specific disease constructs. And so we came up with a measure after hearing this in some of our early trials. We came up with a measure in 2009 with Seaside Therapeutics, which is a situation where we just went in and said to the families, we just want you to rate your worst 3 problems that you deal with your child with Fragile X on a visual analog scale. And this is a visual analog scale where you draw a line between where the behavior is not a problem and it's the worst imaginable behavior. And this actually was the most significant measure improved on our baclofen in this phase, in the phase 3 Seaside trial. But the FDA did not like this because these behaviors were all over the place. Why were they all over the place? Because the important behaviors are different in every family, and so they couldn't be put into categories. So then Seaside said, okay, to the FDA, we'll just have the parents rate domains like anxiety or aggression. And this was a problem because the parents it wasn't really addressing a specific behavior, like he has a tantrum every day when he gets off the bus. And so it was very hard to rate, and the data was kind of a mess. And then Neuron came up with a solution to that in their Fragile X trial, where they sort of said, okay, the parent, we will have the families rate their most important problem, but we're going to have the clinicians map them to categories so that they can then be mapped onto some kind of labeling concern. It's not clear that this solves the issue, but it does personalize the response and allows categorization of responses into categories that you can say the drug works on this area. And I think this measure is really valuable. It addresses the interpatient variability and allows you to kind of target what you think the drug will do, but it keeps the core problem domain assessment. So this is an example also of that trial I just showed you, where we did a visual analog scale, and we were doing a drug that we were using a drug we thought would impact cognition. And so in addition to that result with the Toolbox, we also saw a significant improvement over placebo in a visual analog scale of language and daily functioning. And what did that mean? That means we sat down with the family and said, what are the daily functioning problem that you're working on that limits your adult child with Fragile X the most? And so there was a list. This is just a list of things that families came up with that they really felt life would be a lot better if he could do this better. And then they rated those in the daily functioning scale. And then in language, they came up with things that were limiting in terms of the patient's language. And so this really allows us to look at what is the problem for each patient and each family. And we do have work to do with FDA on this, because they don't like the fact that it's a different thing that each family is rating, and they think it's kind of too narrow. And so we're coming up with ways of getting around this, like using Likert scale ratings, maybe mapping it onto a patient family CGI, and having the family rate like maybe their top 3 problems. But so this is a work in evolution, but it is a solution to the need to really target the problem that the family is having and the patient is having. And just one last problem I want to talk about. And so I've worked in multiple diseases, to name a few: Fragile X, Angelman syndrome, and Niemann-Pick, where the disease is complex and we can't just measure one thing to capture the benefit. It's not like we're measuring a disease that's defined by one symptom. And so these are diseases that we've had to argue with the FDA about the problem with having one behavior or symptom as the outcome, and argue that we need to have some kind of a composite measure or the ability to look at multiple behaviors. And this, for instance, is Fragile X, where if this is data from our Forward Natural History Project, and based on 1,626 patients and about 3,500 visits, we have data on what kinds of limiting behaviors the patient has, and 65% of patients have multiple types of limiting behaviors. So picking any one of these as an outcome measure in a clinical trial is very challenging, and many of them are also on medication, supportive medication for multiple type of behavior. If you look at NPC, there's a huge list of things like ataxia and cognition, dysarthria, dysphagia. These patients have a wide range of different kinds of behavior, and picking the one that might get better in a disease-targeted drug trial is very challenging. Angelman syndrome, maladaptive behavior, seizures, cognition, motor, sleep, these were all things that were flagged in the disease concept model where clinical experts and caregivers were interviewed. And so we have, the key point here is we have multiple symptoms in multiple domains that we have to look at in these complex diseases. And so how are we going to deal with that? And there's a couple of ways that it's been approached, and I don't know that we found the perfect way, but we need composite measures that are going to take into account improvements in any of multiple behavioral domains in Fragile X or other, multiple different functional domains in any of these other diseases. And given that one patient's problem will be something different from another patient's problems, we have to be able to look across all the patients. And so the Angelman syndrome group and the NPC group approached this by developing measures that would look across multiple domains that you could rate. In Angelman syndrome, they have CGIs that are divided into different domains, so you can rate those separately and look at improvement in any one. And in NPC, we have a 5-domain scale where we can look at the 5 most important problems that families flagged as their biggest issues. And then in Fragile X, Neuron tried this composite measure where you actually took 5 different measures and brought them together to give an overall composite score. And we also gave patients scorecards on whether they had improved in 1 or 2 or 3 of these different measures, did a permutation test, and showed significance in their trial. So this was just a novel way of trying to put this all together. But again, I think we still have work to do to bring these measures to the level of validation that is acceptable for drug approval. But these are just some ideas in how we've tried to get around with the need to approach the patients more closely and find out what it is, measure what it is that's important to the patients, as well as approach the variability in many of these rare diseases and deal with the fact that these patients are not functioning at sort of a normal level in society. And that's all I'm going to say. And I want to thank the organizers for inviting me to talk.
Thank you, Jen and Elizabeth. I think this has been a wonderful conversation, and I can see really good comments from audience. So audience, if you have any questions, please feel free to type in the moderation Q&A. Starting off, Jen, I was curious, something that I really enjoyed listening from you is ask patients. And, with your wealth of knowledge and experience working with the patients and also being a patient yourself, I'm curious to hear, when we think about meaningfully designing endpoints or measures or in the research, what do patients value and what have we missed over time? And how do we engage patient organizations, patient advocacy organizations, and FDA to accept that these are important things for the patients?
Yeah, well, thank you for the question. I think that one of the first things to point out when it's like, what do patients want, is to highlight that patients are not monolith, and that's why needing to do the work. I can't speak on behalf of what that would be, but I think it's understanding that oftentimes patients' priorities are thinking about how can they go do the things that they want to do. And so therefore it may be controlling symptoms or allowing them to have, a higher level of functioning than what their condition is currently letting them do. But it might not just be like a, clinical marker. They want to say, I want to be able to do this. And then it's your jobs as innovators and researchers to be able to translate and to think about what might that look like as an endpoint or a surrogate endpoint for something. So that's sort of the first step, is like having those conversations to be able to figure out what they want. And in terms of how do we do this, I can answer how do we do that aspect of it. And then of course, going to talk about what it's like to develop out new measures, but I'm sure that that's perhaps where many people are more versed. But from my standpoint, we do this stuff day in and day out to talk to patients. And this is coming through things just like in-depth interviews, by having these conversations. It can be pulling together roundtables to have a lot of different discussions amongst patients. We do a lot virtually, where we're actually hosting co-design workshops on our platform to be able to accommodate for more inclusion across the places where we're not going to be engaging in in-person opportunities. It allows for people to log on when their kids have gone to sleep or whatever it might be. So there's a variety of different ways. And I also caution people to think about making sure you're picking like the right thing at the right time. I think when people think about how do we get patient insights, they always jump to the surveys. And same sort of feedback and thing I press on with certain like PROs, patient reported outcomes. Just because a patient reports something doesn't mean it's important to them. So we need to go and do the work. But the same thing with surveys, if we're doing on a larger scale, means it's probably more quantitative in nature. And when we think about that, big data can give us a signal, but it doesn't always tell us what's wrong. So we need to marry the two to be able to go to a wider audience, but then to be able to dig in deeper with certain patients and caregivers to talk about what's important.
I love that. I think one of the very important things once we have the patient insights and talk to them and get those insights with them, something that I've seen from the conversations and also something as a next step, and Elizabeth, I think you highlighted this really good, is the regulatory barriers from the FDA side pose challenge in development and evaluation, even if there is great research in the rare disease space. I was curious around what is FDA's process for proposing new endpoints, and or how can we influence FDA now that we have heard from the patients, we have involved them, and we have a great research? What are some factors that will help convince and overcome regulatory barriers?
Yeah, so I think that in FDA wants to understand what the patients want. I mean, I think we shouldn't sell them short. They are interested in patient-derived outcomes. I mean, they're even making recommendations now, like include exit interviews in your clinical trials so that you can really get a more qualitative sense of what the families saw when they were in the trial. And I think that's all important. But then it comes down to that almighty primary endpoint and hitting on that primary endpoint. And that at this point, that's not going to be a family interview right now from where the FDA is. And the problem is that you've got, as Jen mentioned, it's not a monolith. It's more like a field of monoliths that you've got. And you've got patient A who's got 7, 8, and 9, and patient B who's got 24, 25, and 26. And kind of what the field is. But when you have the patient in front of you, you want to measure the things that are important to them. And those things are not going to align with what's important to every other patient. There'll be some aligning and there's overlap, obviously. But in the end, you have to have a measure where you can look across the whole thing. And I think that's been something of an issue that, at FDA, you're used to thinking about labeling and what exactly is this drug addressing. And here we're trying to just address the whole condition. And that's there's a little bit of a disconnect with how are we going to approach that and come to a compromise about how we can measure everything that the patients are telling us and really measure the important things for each patient, even though those things are going to be different for different patients. And that's the correct.
Sounds good. I definitely agree with you, Elizabeth, in terms of like what is important. I think there is a huge alignment that we need to have from the research and the regulatory side. And then it starts with asking the patient first. So I want to thank both of you, Jen and Elizabeth, for this great presentation and insights. And before we officially wrap up, please, audience, please take a few moments to respond to the polls that are being launched now. And we all appreciate your feedback on the session, and we want to ensure that we are able to provide great content and programming for the rare disease community. You can also share your feedback via events@globalgenes.org. And for next few minutes, for next 7 minutes, we'll take a break and we'll return for part 2 to dive deeper onto endpoints and therapeutic advances. Again, thank you everyone for joining, and enjoy your break.
Thank you so much, Ben. It's a pleasure to return to the event today. Today, I'm going to give you a little overview at a very high level of drug repurposing. It's an initiative that we've been working on for over a decade. It's seen a lot in rare diseases across many, many disease areas, including rare diseases. That's a really great option for a lot of rare disease patients, caregivers, as well as all the other stakeholders. So as you've probably all seen and heard over the last 15 months, in early 2020, people around the world saw what we have known and have been working on for over a decade. And that's the fastest way to find treatments for patients with unmet medical needs like COVID-19 is through treatments that are already approved and on the market. It's the same crisis mode that rare disease patients and their caregivers are always living with, and the rest of the world understood the rare disease community pain at that point. So many projects at the peak, we saw over 1,000 randomized controlled trials going on using already approved therapies for COVID-19. Cures Within Reach has been funding fluvoxamine as well as vitamin D. Dexamethasone is something that a lot of people have heard quite a bit about. And Eli Lilly and its baricitinib in combination with remdesivir is a great example of that. How does that work, though, for the rest of the community, the rare disease community, and why is it such a great opportunity? It is a faster, more cost-effective way to get patients these treatments. It is very quick to human clinical trials. It's a lot cheaper to go from beginning to end because there's already the safety profile known. And it is a very well-respected way to not only do follow-on treatments, but also rare disease treatments that have not had any opportunities. And a lot of people consider repurposing not only from opportunities to make an impact, but bridging time before new therapies can be brought onto the market. It's also a really good alternate bridge to new therapies. I want to make sure that everyone has a good understanding of some definitions. Repositioning is what we think of when industry might shelve an indication, either at phase 1, phase 2, or at phase 3, that doesn't make it all the way to regulatory approval, versus repurposing, which is when a regulatory agency, the FDA, could be the EMA in Europe, it could be Canada, it could be any agency around the world, approves it for a specific indication. It's safe, it's effective for a specific disease, and that's called repurposing. And Cures Within Reach also differentiates between philanthropic use and commercial use. So commercial use is when there is a regulatory path and an industry player, whether it's a very large player or a very small player. It could be a startup, it could be a player like Lilly or Novartis, who brings it all the way to the FDA, for example, for approval as a second, third, or fourth indication. When there is commercial value, there might be intellectual property, there might be market exclusivity, but it's different than philanthropic value. And that's when we look at it from the patient's perspective. It may be a generic, there may never be an opportunity for regulatory approval, But patients don't care. If it works on a patient, it's a generic. There's a clinician or a researcher who publishes in a peer-reviewed journal. It's statistically significant information. It's reproducible. And that's allowing clinicians anywhere in the world to use it off-label. And that is a wonderful way for patient impact and what we would consider philanthropic use. So today we're going to look at repurposing, and we're going to look at either philanthropic or commercial value. Since the Cures Within Reach, we're indifferent to either philanthropic or commercial value. At a very, very high level, Cures Within Reach for over a decade is leveraging this opportunity for repurposing to quickly impact patients, regardless of philanthropic or commercial value. We're disease agnostic, we're geography agnostic, we're institution agnostic. For over a decade, we've invested more than $6 million to catalyze first-in-human pivotal studies, and our researchers that have funded has then leveraged and raised follow-on funding for follow-on trials of over $67 million. We have 33 projects ongoing right now. 42% of these are in the rare disease community. 5 have been, that are ongoing now, have been winners of a past Cure Accelerator Live for Rare Disease event that we held yesterday and announced the winners today. So as of this morning, 2 more will soon be joining this group in the rare disease community. In addition, we have 24% of these projects that are in the pediatric space. And it's a great example of repurposing. What I really want to spend more time, though, this afternoon, is looking at repurposing considerations for patients, for caregivers, for the patient advocacy community. What are the advantages? What are the challenges? So a handful of things to keep in mind. I'm going to kind of run through this list quickly and then use some of our success stories and ongoing projects as examples to more food for thought for the attendees. So could it work in your disease area of interest? The answer is likely so. Whether it's a rare disease, an ultra rare disease, it could be a genetic related disease. Very frequently, repurposing may be something to consider, certainly not the only thing to consider, but it could be something to consider. Enrollment in first-in-human pivotal studies can be very small. It can be 5 to 20 patients for a first-in-human phase 1, perhaps phase 2A trial. It doesn't need to be a lot of patients. Sometimes it needs to be more, but oftentimes it's very small patients. From an intellectual property standpoint, there might be intellectual property, but there might not be. And that's a TBD, but it's not relevant if you're looking at that first-in-human trial necessarily. It doesn't need to have intellectual property value due to the ability to look at off-label use as an opportunity for impact. As I already mentioned, there's a known safety profile and all the related information that goes with it that allows the researchers, the research institutions to move quickly to first-in-human studies. It can, like I said, it can be done in almost any disease. And the goal is to show clinical impact, show that you're making an impact in patients, either quality of life, in progression of the disease. You may not know the mechanism of action right away, but from patients' view, it's not necessarily why it's working. The fact that it's working is what's the most relevant. In the United States, there's something known as the 505(b)(2) for a regulatory path. There's a lot of other incentives outside of the United States, as well as even within the United States for orphan disease designation, market exclusivity, and other opportunities. Another advantage that we are really interested in and curious about over the next couple months and years is the opportunity that artificial intelligence and machine learning can bring to repurposing. There's many organizations do a lot of really interesting work in taking databases and libraries and saying, we think these 5 therapeutics already on the market may have an impact in a specific disease. And you're really reducing risk and strengthening the opportunity to make an impact. All of those still need to be tested in human clinical trials, and that's where Cures Within Reach sits. So we really see a lot of the AI and machine learning initiatives as a really great pre-step to where Cures Within Reach ourselves gets involved. On the flip side, there are some challenges and some disadvantages, so to speak, to be aware of. Not necessarily they can't be overcome. They certainly can be overcome, but just to keep in mind. One is that many research institutions, not all, many research institutions consider repurposing as not innovative. It's not a new drug. It's not a new class. It's not going to be a billion-dollar blockbuster. But to patients and to caregivers, they certainly don't care about that. The challenge is getting the institutions interested and engaged and involved to see it as innovative when you're going to solve patients' concerns. It can be hard to find funding, especially when there may or may not be any commercial value. At the same time, the key is to seek out researchers, clinicians, key opinion leaders who are dedicated to your disease-specific area of interest. More than likely, one of those groups is going to have repurposing as an initiative, as an opportunity, and you really want to partner with those key opinion leaders, with those research institutions. I'm going to walk through a couple examples that I think highlight the opportunities that patients and their caregivers can have when you're looking at repurposing. The first example I'm going to share is a winner of our 2019 Cure Accelerator Live for Rare Diseases event. This is coming out of St. Jude. It's repurposing ruxolitinib, a blood cancer drug that's currently on the market, and she's repurposing it. Dr. Nichols is repurposing it for a rare immune disorder, HLH. It's a great example of where patients can get involved, looking at a very specialized team like St. Jude to improve these options. And the St. Jude team engaged pharma with this clinical trial and successfully got pharma engaged to donate some drug. Only 10 patients are in this pilot study that Cures Within Reach funded. Of course, the long-term goal is a larger phase 2 study, but that is a great example and it's ongoing at St. Jude today. Another example would be something that actually was one of the original projects that allowed Cures Within Reach to change our focus to dedicate ourselves to repurposing over a decade ago. And that's a very rare disorder called ALPS, where we had a researcher out of the Children's Hospital of Philadelphia, so right in UPenn Orphan Disease Center's neck of the woods, who's testing sirolimus. And Dr. Tichy used sirolimus, a rare, an approved transplant drug, to make an impact in this. And this is a great example of a philanthropic value. So this is a generic. It was a very small study that was very successful and immediately saw an impact in these patients' lives and these kids' lives, so much so that 5 additional diseases in this space were tested, and it's being used off-label even today. The final example I'm going to share with you is another one from our early days. I think we've all heard about thalidomide that was approved for leprosy. We funded a Mayo Clinic researcher early in his career, a young investigator at the time, looking at using it for multiple myeloma. It got on the market, great example of some commercial value, finding a researcher with a passion who really wanted to make a difference. A great example of what industry engagement can certainly do as well. In closing, I want to say I'm looking forward to the conversation. Starting with small pilot trials is a great way for patients to get involved, find a key opinion leader and a clinician as a partner. And it's really important to think about the early initiatives to catalyze the follow-on funding. Again, looking forward to the discussion. I'm going to turn it over to Dan O'Reilly for the next conversation.
Thank you, Barbara, for the kind introduction. So today I'm going to give you like a broad overview of what are oligonucleotides. You may have heard of them. It's called as ASOs or antisense oligos. So to do this, I have to explain a little bit about DNA. Now, DNA is like information storage for all our genetic information, and you can think of it as like a recipe book where you can produce these nice like cakes here. In fact, in the cells, it's a bit more complicated than this, where you have a process where the DNA gets made into something called RNA and then into a protein, which the protein essentially performs all the functions in the body. Now, when there are errors in this genetic information or mutation, this leads to a poor cake. Now, no one wants to eat this cake, and this is what happens when there is a genetic disease. You get a mutation in the DNA that results in a downstream effect of causing problems at both the RNA and then the protein level, which result in a disease. Now, oligonucleotides are really innovative because they can target these RNA or to help block the production of the protein of these specific mutations. Now, to explain how they work, we have to actually talk about the structure of DNA. So some of you might recognize the double helical structure of DNA on the left. It has 2 strands, but essentially they're made up of 4 bases, A, G, T, and C. Now, what's really great about DNA and other nucleic acids is if you know the sequence of one of the strands, you can work out the sequence of the other by 2 rules. One is that A pairs with T, so if you have an A on the first strand, there's a T on the other. And the second is that G pairs with C, so if you have a G on the first strand, on the opposite strand, there's a C. And I have a small sequence here to highlight this. So we have the sequence TCGA, and in the opposite strand, the other sequence will be A, because A goes with T, and then G, because G goes with C, and then CT, because the same rules apply. Now, thanks to the Human Genome Project, we actually know the sequence of all the DNA and the RNA in your in the bodies. And now, to explain what RNA is, we can compare it to DNA. Now, it's actually very similar to DNA in the fact that it has the same bases. It has A, G, C, and it has U, but for all our intensive purposes, it's the same as T. But the key difference is that RNA is single-stranded, whereas DNA is double-stranded. And this means that we can use the same ATGC rules and make a sequence of those bases, a small short piece of DNA or an RNA, to target the RNA that is produced in your cell and hopefully stop the disease. Now, antisense oligonucleotides work by 2 pathways. There's the steric block antisense oligonucleotides, where the synthetic piece of DNA just binds to the RNA, and then you can actually manipulate the protein structure this way. And on the right is a gapmer, which we don't need to go into why it's called gapmer for now, but all you need to know is that this way it actually stops the production of the RNA and stops the production of the faulty protein. Now, there is another class of oligo-based therapeutics, which is short interfering RNA or siRNA. Now, these use the same ATGC rules to target any RNA we want, but it uses a double-stranded piece of RNA, and it loads into this risk, and but the outcome is still the same as those gapmer ASOs I just showed you, where it stops RNA production. Now, there are about 10 approved therapeutics for rare genetic diseases, based on the 3 methodologies that I showed you. But the question really is, well, which type of the 3 ASOs is right for your disease, and how do you go about deciding which one is the right? And it comes down to go into this a bit more. I'll talk about you need to know your sequence mutation first. So you have to sequence your mutation and actually know what it is and where it is in the genome. The second thing that you need to understand is actually the biological effect of this mutation. So what happens downstream? So what happens at the RNA and then the protein level that causes the disease? And so this I'll go into a little bit more detail for briefly using Pac-Man as an analogy, where if we can imagine our proteins are Pac-Man and its goal is to eat the yellow dots. So now one biological misfunction you can think of is that if you have a mutation that leads to the protein actually having the same function, but also having a toxic function as well. These are called toxic gain of functions. And so now we can imagine the Pac-Man has these razor sharp teeth, which means that he can still chew his balls, but he can't work. He does other effects and even damages himself. So actually how researchers necessarily go about using oligos is to block these and reduce the amount of this protein using the gamma ASO or siRNAs that I mentioned earlier. An example is like Huntington's disease, where you have this toxic gain of function, and researchers are currently working on these types of oligos as a treatment for that disease. Now, another type of biological outcome is a loss of function. So here what happens is the mutation produces a protein that doesn't work. And in this case, the Pac-Man's mouth is shut, so it can't actually function properly. And so what researchers can do is bind an oligonucleotide to using steric block ASOs to the RNA at the pre-RNA level, which changes the genetic information that actually goes into the RNA and produces a protein that's hopefully functional. An example of this is Duchenne muscular dystrophy, where they actually produce a semi-functional protein so that patients get a less severe disease. Now, DNA and RNA are like, if you just inject it into your body, it's actually a poor therapeutic outcome because it elicits the immune response, can have off-target effects, and gets degraded very quickly. Now, to overcome this, scientists have developed chemical modifications to the DNA or RNA to actually make it a better drug. And so I'll just go into a little bit more detail about this. And so for this, all you need to know is that the DNA I showed you earlier is actually made up of small building blocks that is represented here. And this building block has a phosphate in blue, a sugar in red, which is the same sugar that you put in your tea or coffee during your day, and then the base, which is the AGCT that I showed you earlier. Now, again, on this slide, there's a lot of chemical structures, but all you need to know is that on the left is the top left is the DNA structure, and all the other structures, the red is highlighting the difference from that natural structure. So you can see that some of the modifications are subtle, where there's like an oxygen to a sulfur switch, or some of them get really different from the natural structure, but they're all there to help the DNA or RNA become a better drug. So I hope that this overview has given you some details on what oligonucleotides as therapeutics and how to go about using them. I would just like to say that there is you can get more details on this at the Oligonucleotide Therapeutic Society, where we have regularly hosted patient advocates, and we have a briefing document designed to actually really go through all details that you need to do when thinking about making oligos for a rare disease. We have a meeting this year, and there's discounted membership for patient advocates. I'm currently the co-chair of that meeting, so please, anyone, come along and talk to us. We're a great bunch of people. And with that, I'd just like to thank my lab and the OTS team.
Thank you. Hello, everyone. Nice to be speaking with you. I'm Yael Weiss from Ultragenics, and I'm going to talk to you a little bit about gene therapy today. I'm going to leverage Dan's previous presentation, so I could skip some of my first slides, and we'll have more time for discussion later. And also tell you that I shamelessly took these slides from Ralph Schmidt, and I'm going to use slides that he showed earlier this week. Not the whole deck, but some of it, and hopefully those who weren't there will benefit from these slides, and those that were there don't mind seeing his slides again. So Dan told you a little bit about DNA and RNA, and instead of repeating this explanation, I'd like to talk about gene therapy in context of DNA and RNA. In the case of gene therapy, as you all know, we are actually at the moment replacing missing proteins. So this would be the classical gene therapy. The newer platforms that are coming out and are being used now are gene editing platforms, and it's different than gene therapy, although the same carrier might be used for both. Looking at this slide, you will see that in the case of a mutation in the DNA, there's usually lack of an mRNA, or the mRNA is making the wrong type of protein or not making enough protein. And what we do with gene therapy, if you look on the right-hand side, is we actually add the right DNA, the non-mutated DNA back into the cells and enable the cell to make the protein that's missing. So that's the context of gene therapy. Now, Ralph used this slide, which I really liked, to demonstrate the difference between the actual deliver, the FedEx of the gene therapy, so the capsid or whatever will contain your gene inside, and then the payload, which is the actual gene that we're trying to replace. And I'll focus on the delivery vehicle because I'd like you to understand that the two major types of delivery vehicles that are used in gene therapy, and what the difference is between them. And when you're thinking of whether you're going to do gene therapy at all, and if you are going to do gene therapy, which capsid you want to use or which delivery vehicle you want to use, you need to understand the differences between them. So the two major delivery vehicles are both viruses. As you know, we all have the COVID experience now. Viruses find their way into the human body and inject their own genetic material into our cells and create havoc in our bodies. We are actually using similar viruses, but taking out the bad parts and instead putting in the genes that we want the virus to make. So we're actually harnessing something that normally can be detrimental to our body into something positive. So the first virus that we use is used pretty commonly is lentivirus, which is mentioned here as LV. And lentivirus is a virus that is used to transduce mostly blood cells. The reason that it's done in blood cells is it's a virus that actually inserts itself into our DNA in our cells. And the reason that we want to monitor it and do it only in blood cells is because we want to make sure that the gene that we're inserting didn't go into somewhere bad and like maybe cut through a gene that we actually need and will create some issues by actually starting to express a protein in a place that it's not supposed to be. So for lentiviral therapies, the cells are taken out of the body, just like when we do blood cell transplantation. The cells are taken out of the body, manipulated out of the body, and brought back into the body. And in the cases where we usually use lenti is for blood disorders like sickle cell or beta thalassemia, but there are some recent reports on neurological diseases that benefit from lentiviral gene therapy, such as Angelman disease. But these are new publications, and these haven't been in the clinic yet. I think that UC Davis is starting a trial for Angelman with lentiviral drug delivery, gene delivery. The second delivery vehicle is AV. This is what we use at Ultragenyx. It's also a virus that has been manipulated to bring its DNA into the body. It doesn't integrate into our DNA. It sets up its own replication system inside the cell and continues to make protein. But this is what actually enables us to give it directly because we know it's likely not going to create any damage into our genes. AV is used in many organ systems, in many tissues, in the eyes, in the liver, in the muscle, in the brain. And we already have some products out in the market for SMA and for retinal diseases. And we're going to learn more and more about these platforms, and as more clinical data comes out about the efficacy of delivering to specific tissues, and also about the safety of these platforms. People are trying to deliver genes using lipid nanoparticles. At the moment, there isn't enough evidence about the long-term efficacy of these platforms, but we will learn more as these platforms develop and more data comes out. So I was asked to talk a little bit about the pros and cons of using gene therapy. So first of all, not every gene is eligible to become a gene therapy. In some cases, the genes are too big. And unfortunately, the packaging system of these viruses only allows a certain type of genes packaged into them. So those that are too big, in many cases, unless there's a way to shrink them, like what's done in Duchenne, there's a mini gene, but it's not necessarily the case for every gene. If you can't shrink them, you can't put them into AV. And then think about treating very young kids, treating an organ like the liver that is constantly growing, and the effect of the gene therapy will be lost over time. So treating very young kids with a growing organ might also be a limitation till we learn how to redose, and we still don't know how to do it because the body makes antibodies to the virus. And this is the third limitation of gene therapy. The third major limitation to gene therapy is that the body makes antibodies to the virus. So first of all, some people were exposed to the wild type virus, to the virus that wasn't manipulated even before they knew they were sick or before they knew they were going to be treated with gene therapy. And the moment they have antibodies to these viruses, the moment the virus will be inserted into their body, the body will try to fight it and create an immune response. So some people cannot be treated even before they were treated once with gene therapy. And definitely those that were treated once with gene therapy make antibodies to the capsid, to the virus. And they also cannot be treated again because the body will always recognize the virus as something negative and try to fight it. But many companies are working on ways to redose, either by tapering the immune response or trying to administer drugs at the same time that will reduce the immune response as the drug is injected into the body, and hopefully in the future we'll be able to redose. I think I'm running out of time, even though I have a lot more to say as usual, but I'll be happy to answer any questions in the Q&A session. Thank you.
All right, thank you so much to Barbara, to Dan, and to Yael for the presentations that we heard. Those were absolutely wonderful. Beautiful descriptions of some very complex things. And we got some great questions that came in from the audience as we were listening to those. Dan, your Pac-Man incorporation was a huge hit. So I'm always an advocate for incorporating 1980s video game characters. So I think a big question that kind of runs as a theme through what we were hearing, especially through Dan and Yael's presentations, is when it comes to it's a question from Mary Hanada. Is it more practical to treat each variant of a gene mutation as a treatment, or to maybe change the entire gene so that you encompass all of the different mutations together? And I think, Yael, you kind of answered that in your explanation that with gene therapies, you're somewhat limited in the size of genes that you can use. But Dan, perhaps maybe taking a more targeted approach with ASOs could help answer that as well. So just like to hear your thoughts.
It's always an interesting question because from a company's perspective, you want to develop a therapy in which you can treat as many patients as possible. So if you have a way to do it, I think it would be the preferred approach. But obviously, that's not the case. And for every type of gene mutation and every type of gene, so we would look at ASOs as a solution, and Dan can talk a little bit about that. And then there might be cases where we can treat a lot of patients because there's a common mutation, and there might be cases that you can't, and then you want to address that.
Yeah, thanks, Yael. I think ASOs do provide a way of treating multiple different mutations because you could target multiple different RNAs. But even then, depending on the amount of mutations, it gets very, very complicated. For example, in Duchenne muscular dystrophy, there is multiple mutations in different parts of the gene. And so even then, you're talking about to treat everyone from one specific cocktail of ASOs. That's a lot of ASOs that you'd still have to design. And even for like ALS, I think there's multiple genes that cause ALS. So again, that's multiple ASOs. And so it does get complicated there to do a targeted approach versus a non-targeted approach.
Sure. So a question comes in from Jin Kao. I think he's asking in the context of gene deletion or duplication, but I think that it could be broadened to a more wider kind of question. Is it generally a more simple approach or a more attractive approach to control gene expression than it is to try to edit the transcript itself in terms of ease or safety when looking at these technologies?
That's a good question. The answer is probably it's safer to do the ASOs because they're approved and the regulatory body understands the side effects from that. Whereas for editing the actual RNA transcript, it's still at a very pre-translational level where no one's approved that yet, and there's not the full understanding there of the off-target effects. So I'd say it's still the ASOs is still probably the best way to go.
I agree with Dan, and I think that manipulating expression is still unknown because the body has very tight ways to regulate expression of genes, and to try to make it deviate from what it naturally does might be challenging and also might be difficult to control. And I think we'll learn more and more about these because there are many companies and scientists that are trying to upregulate haploinsufficient alleles, and we'll see if this is successful and leads to the results that we're interested in seeing.
Awesome, thank you. Last question that we'll ask here, and I apologize to everyone who asked so many beautiful questions. There's just not enough time in the day, but we will do our best to follow up one-on-one afterwards. So for the whole panel, if a patient advocacy group or a group of patients has a therapy of any kind that may help the population, what are your thoughts on best first steps on taking it to the next level, working on engaging researchers, starting a trial, seeking funding, all of those sorts of things? It's a loaded question, but I think it's a common one, one that everyone asks themselves at some point in time. So what does this panel think would be the best starting points to take that first step on that long journey?
I'm happy to start at a high level. I think patients and really the entire ecosystem needs to look at it from a short-term opportunity as well as a longer term. So, through off-label use, through case reports and real-world evidence, you could start with a clinician who's prescribing something. If it's already on the market, for example, you could start using it personally pretty easily if you find a clinician that's working with you to prescribe a particular medication that's, again, already safe and effective. From the repurposing lens, you could absolutely go that route, and it might work for you personally. It might work for a small group of patients. There's a lot of ways to share data that comes out from that. At the same time, though, that's not going to impact patients on a broader scale. It's not going to impact patients on a global scale. That requires randomized control studies and that are peer reviewed and reproducible. That takes a lot more effort, where you need to find a clinical research site that's going to work on that with you in partnership with you. gets funding, you need funding on that. So just think about it from, there's the short-term immediate impact. You want to impact yourselves or whoever in your ecosystem has that disease, but there's also a longer-term impacting patients broadly, which needs to be a randomized control study.
Talked about it a little bit on Wednesday at the pre-conference workshop, and I think the advice that we gave was to create a toolbox of everything that you need in order to develop drugs, whether it's cell lines or mice that have the relevant mutation, anything that, understanding the gene, understanding the mutations, understanding the biology, and put in place an advisory capacity. It doesn't have to be a long-term advisory board, even a one-time scientific board that will sit and go through all the information with you and try to help you determine what the best approaches are, whether it's repurposing a small molecule, looking for a new small molecule, ASO, gene therapy. And again, we talked about the fact that if you're a very rich foundation, you might want to try everything. But if you have limited resources, you want to sit with experts that understand the platforms and understand your gene and its biology and match the right modality to your disease.
I agree. Understanding the biology and having people around you that know the science and can advise you correctly is like really important. I also think to start getting those people, go talk to people at like global genes events, even come to like the OTS for oligos, like go to conferences where these people meet and just talk to scientists, because many are willing to like help or get involved. In fact, like I know many academic labs that are helping try and develop oligonucleotides for rare diseases. And so they may or may not be willing to help.
And we also invite you to join our Rare Entrepreneur Bootcamp that will hopefully take place in October, face-to-face. And we spend 3 days discussing drug development and different approaches to the business of drug development and teaching you how to do it. So, and from Global Genes, we'll be happy to give you additional information on this.